Major epigenetic tool for refining glioma classification. DNA methylation profiling also has potential application in classifying other tumor types, including sarcomas and carcinomas. Diagnostic and therapeutic implications are important. Professor Hoffman, the speaker you will introduce, will also address emerging methods for rapid DNA methylation-based classification that could potentially be applied in intraoperative settings.
So I don't know if I have to speak in English or in French. In French? In French, I don't know. Bon, c'est un grand plaisir, un honneur d'accueillir le professeur David Capper, qui vient donc de la Charité, l'hôpital de la Charité à Berlin. Par hasard, j'ai travaillé avec David Capper par Internet sans le connaître, il y a 10 ans. Donc, grâce à la découverte par David d'un anticorps qui a rendu beaucoup de services aux pathologistes, un anticorps qui est dirigé contre une mutation sur un gène qui s'appelle BRAF, qui est extrêmement fréquent dans sa mutation sur les mélanomes malins, mais pas que, également dans les cancers du côlon, dans les cancers du poumon, dans les glioblastomes, les gliomes. Et bien, très gentiment, David m'a envoyé un petit alico d'un anticorps extrêmement précieux qu'on a développé ensuite sur nos échantillons au laboratoire. Et on a publié ensemble, et hier soir, j'ai fait sa connaissance face à face, et non plus par Internet, ce qui est beaucoup plus agréable, surtout autour d'un bon repas. La communication est importante, c'est pour ça qu'effectivement dans l'intelligence artificielle, l'empathie du médecin est quelque chose aussi de très important. Il ne faut pas couper la communication humaine, c'est un peu l'enjeu actuellement. Et grâce donc à ce repas, j'ai recontacté David. Donc au-delà de ça, David est neuropathologiste, donc pathologie spécialisée dans les tumeurs cérébrales, issu de l'université d'Heidelberg, et maintenant à Berlin. Et David a énormément travaillé, fait des magnifiques publications sur la classification des tumeurs cérébrales, en ajoutant un point très important, la méthylation. La méthylation, donc c'est une partie de l'épigénétique, vous avez également les micro-ARN dans l'épigénétique par exemple. Et la méthylation finalement est très importante parce qu'on peut trouver de nouvelles cibles thérapeutiques grâce à des profils de méthylation dans certaines tumeurs cérébrales. Donc c'est un pas de géant dans la médecine personnalisée des tumeurs cérébrales dont on connaît le prognostic extrêmement défavorable. Et finalement, c'est la première fois qu'on avait affaire à une modification des classifications histologiques en incluant des profils moléculaires. Et l'OMS a donc intégré les profils de méthylation des tumeurs cérébrales dans sa classification histologique. Donc les pathologistes sont obligés maintenant d'intégrer la biologie moléculaire et en particulier dans les tumeurs cérébrales les profils de méthylation qui sont rentrés en routine diagnostique. On ne peut plus faire un diagnostic de cancer du cerveau sans intégrer les profils de méthylation, et David Capper donc a vraiment boosté cet axe de recherche qui est maintenant rentré en routine, et on peut l'en remercier car grâce à ce type de développement de projets de recherche, on améliore la survie des patients dans une pathologie qui était jusqu'à présent extrêmement péjorative, qui le reste, mais c'est le premier pas vers la médecine ultra stratifiée des tumeurs cérébrales. Donc merci beaucoup David d'être venu de Berlin nous faire cet exposé. J'espère que tu as tout compris. Oui, tout.
So, dear Paul, thank you very much for these very, very nice words. I think I understood most of it, or at least a large part of it. I'll switch to English now, and I'd like to first also thank you, Isabelle, very much for this very kind invitation here. So, I will talk about DNA methylation, and you will also see that some of this will also attach very nicely to the previous talk of artificial intelligence. Because what I will demonstrate here now is that what has changed neuro-oncology a lot over the last years is that we have found a new code that is very well suited for classifying brain tumors. This is DNA methylation, and I'll show you a bit of background what that is and how that functions. But the biggest advance in this field is in fact that these profiles are then analyzed by machine learning tools, kind of a simple form of artificial intelligence, and that this is very far advanced in routine diagnostics in neuro-oncology. Just very few words on potential competing interests. You also, I think in one of the last questions, just heard that regulation of artificial intelligence is a big problem at the moment, and that integration into medical, practical medicine kind of requires a lot of regulation and adherence to regulation. Therefore, most of this research was done by the German Cancer Research Center, and it has now very recently decided that this has to kind of be handed on to a company which was founded where I'm a co-founder and a shareholder, which is called Heidelberg Diagnostics. So the main focus of this company will be to battle kind of, or to fight out these regulatory issues, kind of that an artificial intelligence faces in our medical system, and to try to make a real medical product by European law out of this. I think the other conflict of interests are of little relevance here. So this is an image of how I was trained on pathology, and which I sometimes show just to give you an impression of what different types of brain tumors look like. And I started my training now 17 years ago. And in principle, what we had to learn back then is that there are different types of brain tumors, that they can look differently, that they have very different names, that they have very different prognosis, and that it's very important for us to differentiate these. And the main tool that I was trained on is the microscope, which is still my daily instrument, I have to say. So I look through the microscope every day, and I try to give very specific names to these tumors so that the oncologist then has the possibility to act or to give the patient the correct treatment or the best advice of how he should plan kind of the future of his life. We have some tumors that are very benign in the brain. We have many other tumors that are very highly malignant in the brain, and it's important for us to separate out these different tumor types. So over the last almost 150 years, a lot of this information has fed into WHO classifications of various organ systems. This is the illustration here of the brain tumor WHO classification, which now separates over 120 different types of tumors. And for many of these tumors, we still have the problem that histology doesn't have very sharp boundaries, meaning that many of these entities have overlapping histologies, that we also have many subgroups that can be very hard to identify, and that in principle we're still working on, or that it would be very desirable to have a better molecular classification scheme that allows a reproducible and global diagnostic standard across neuro-oncology. A lot of these things have been addressed over the last years. Molecular pathology, that we will also hear in one of the following talks, has helped us with many of these questions, and today I would like to focus on the contribution of DNA methylation in this field. As I said, that has been very far advanced in neuro-oncology, has been accepted by the WHO classification. An image of DNA methylation also made it to the cover of the WHO classification, and it has become a very integral part of classifying brain tumors, and I'll try to show you a bit why. So DNA methylation, as many of you will not work with this on a daily business, is part of the regulatory machinery of expression of every cell that we have in our body. So it's a very natural process, and it is very powerful, in particular from the differentiation of an embryonic stem cell to a kind of fully developed cell of the body. And it allows us to have different cell types, like an astrocyte in the brain, like a gut cell. And this is kind of that these cells are very different, has to be recorded in the system, and DNA methylation is one of the important parts kind of this regulation. DNA methylation, in most cases, takes kind of its role not within the coding region of a gene, which is here depicted down here, of a kind of made-up protein here. but it is rather outside of the gene in regulatory regions, mostly in promoter regions, and if methyl groups attach to the DNA in these sites, that will block transcription factors from binding in these regions and will then switch off proteins. So it's a very simple process, absolutely normal, happens in every of our cells, and is very, very central to the development of a multicellular organism. On the other hand, we have realized that tumors also have very distinct DNA methylation profiles. If we look at these in a kind of throughout the whole genome, and we don't yet fully understand why this is the case, we assume currently that it will have something to do with the DNA methylation changes throughout progression from a stem cell to a fully developed cell, and that at specific time points, cancer cells can take over these cells, so kind of in the course of the first initial mutations that happen in these cells, and that likely the DNA methylation that we then see, if we look at a tumor type in the end, is more or less a recording of the cell state at the time point that the initial mutations took over the cell. So that's a theory at the moment, will have to be demonstrated in the future, but that is at least what we're working with. DNA methylation happens at approximately 20 million positions in the human genome, so we have a lot of data that will be possible. And one of the big advantages in this field was that a company that I'm not related to here, Illumina, produced a very high quality methylation array that allows us the interrogation of 850,000 of these CpG sites in a very easy high throughput manner. Meaning that from a diagnostic sample or a research sample, we can very easily record for 850,000 of these individual sites, is there a methyl group at this site, is in yes or no. And that is of course a great baseline to do calculations, to do mathematics or machine learning to recognize patterns. The second big step forward of this technique was that in comparison to many other high throughput methods, like RNA profiling, etc., this works on formalin-fixed paraffin-embedded tissue, which is these little blocks that pathologists usually work with, which are very easy to kind of to store, to hand over, and also to archive, meaning that the introduction of this technology allowed us to go back into the archives of pathologists for 20, 30 years and look for specific specimens that are often very rare that you need to kind of enrich your reference cohort with if you want to classify the whole full breadth of cancer. This is what a very early classification tool looks like. In fact, the first one that I know of was created or published in 2013 and shows here the comparison of for a specific brain tumor, which is called medulloblastoma, which is a highly malignant tumor, a tumor of children, mostly in the cerebellum, which had been known that it is at least consists of 4 different disease types that could be tested by RNA expression profiling, which are color-coded in these different colors down here. And this is almost 200, or a bit over 250 samples, and every tumor is in a thin line here. And you can see that they separate very nicely by RNA expression. What is shown in the heat map at the top here is that you can also use alternatively DNA methylation profiling. And here in red you would have sites that are highly methylated, so have a strong methylation signal. In blue you would have sites that have a low methylation signal. And what you maybe can appreciate here is that for every of these colored classes down here, you have something like a fingerprint kind of pattern of lines of red and blue that represent kind of the molecular profile of this tumor. And back in 2013, I was still in Heidelberg at that time in neuropathology, and we discussed these results here and decided that if it is possible for a single tumor entity to separate kind of 4 very specific subtypes, it should also be possible to classify all types of brain tumors if we had a very big reference bank where all types of brain tumors would be included and we would have the methylation profiles of all types of brain tumors. And in principle, not even all types of brain tumors, but all types of cancer. It could be envisioned that we may have the possibility to classify something like this and to generate an alternative to the microscope. Because testing like this is completely independent of a microscopical evaluation And using such a system, we could for the first time evaluate also how good is the microscopic evaluation in total. That was done, and over the next 5 years, we created a reference cohort incorporating all types of brain tumors that we could get our hands on. This depiction here, every little dot is a patient sample, and in this graph here, the DNA methylation information, so this very complex information, is reduced just to a single dot. And what you can see is that dots of the same color, which have the same histological diagnosis, group very closely together. And in principle, this is a very simplified map of DNA methylation profiles across different types of brain tumors, where you would have a specific site. This would be, for example, the astrocytomas at the top here. This would be the glioblastomas down here, where you have different regions on this map that belong to different types of cancers. And this map can then very easily be used for machine learning. If you have a new diagnostic sample, you compare the profile to all samples that you have in such a map, and will get a result out of this. And that is, in principle, what was done for the diagnostic testing of this. So you can then use such a reference cohort, use a test sample, generate the same data type, so DNA methylation data, and then compare it to such a system. And you can also do inherent testing, so how good is the system running if you, for example, divide your reference cohort into two pieces and then generate a new classifier and run this through. So you can do mathematics on such a clean test set, and you can define that the misclassification rate of such a system would be approximately 1%, which is of course not optimal. You would still say, okay, 1 in 100 would get a wrong diagnosis, but the big question at that time was, how does that compare to pathologists' diagnosis? If we compare them in a blinded fashion, so how high would the misclassification rate between these two systems be, and how would we think which system would actually win over the other system? That was done in a blinded fashion in Heidelberg, which is a big expert center for the classification of brain tumors, for over 1,000 tumors in a blinded fashion. And to make this very short, what came out of the system is that it doesn't recognize 12% of the tumors that it is being fed with. It just tells us, I do not recognize this tumor, it doesn't belong to my reference cohort. And that was the first big interesting thing. We recognize that we are seeing maybe much more tumors that actually don't belong to established classes than we expected. So that was an important number, and I'll show a few things on this group in a second. And the other important number was again 12%, where the system gave us a different information, and the cases were reviewed, and it was then also seen that that indeed, kind of by looking at a kind of additional standard analysis and very in-depth molecular characterization, indeed the system corrected us as pathologists in 12% of brain tumor diagnosis. Some of these big differences, kind of with switching completely from classes to benign to malignant or the other way. Others were more subtle changes kind of within kind of closely related classes. But a substantial number of these was indeed seen as highly medically relevant. That is also kind of the data that in the end convinced the WHO after it was reproduced also by other users in such a setting here. One of the nice things about DNA methylation is that it can very easily be transferred through the internet, because it is not being considered as identifiable as standard genetic data is. So the internet portal was created by the German Cancer Research Center, where you can upload your data and then for free generate kind of such a classification report. And that has been used very, very heavily over the last years. So it has seen over 100,000 website uploads of diagnostic samples here. And what the German Cancer Research Center does, if uploading people are okay with this, and if this also adheres with their local laws, is that they ask for a data donation. And by using this, a massive data set has been brought together. So starting from the initial classifier that just had around 3,000 brain tumors that were included, there's now a data set that is over 125,000 methylation profiles of human brain cancers that kind of are all in the same place, which is far more than any highly experienced neuropathologist or pathologist could ever see in their lifetime. And this dataset was then used to identify new brain tumor classes. because it's of course obvious that within this data that is now all of the same quality, we are now able to identify very specific new groups that no pathologist before could have ever identified. And already in the WHO classification of 2021, a substantial number of new tumor classes, I think it's 13, were identified by DNA methylation profiling and have been introduced into the official canon of the WHO classification. Just to show you one example how that would work, because we're still doing this and identifying further new tumor classes, is that we would look at this map, would then kind of in the color codes the different known tumor classes, and we would then identify little islands kind of in this group here that don't represent a known tumor class, would then go into detail into these groups, would then try to identify as many of the samples of this, look at them histologically, but then kind of do genetic testing, for example, in this very, very small group here, which is just one of 7,000 brain tumors kind of that come into such a system here. We identify that there's a very, very highly recurrent genetic alteration that is not described in other brain tumors, and we would then kind of characterize such an entity also from a clinical perspective. And this, for example, is a project that a doctoral student of mine is currently working on, where we are pretty sure that in fact we really have a highly, highly rare new subtype of very benign adolescent tumor types that was just not recognizable before because it is just too rare. One further thing about DNA methylation is that we have found a common language that crosses boundaries between different countries for diagnosing cancers. So we've tested this, but there's also further publications on this here, that we can use DNA methylation profiling if everybody uses the same kind of algorithm in the background, then irrespective of where this data is being generated, you have a massive overlap of results kind of between these centers. We tested this for very specific tumors, where we just did a centralized DNA extraction and then sent out blind DNA samples, so without any attaching information, to 12 European centers, also here in Belgium, and then asked the diagnosticians to come to a full diagnosis just from this DNA sample. As I said, it was quite specific histological subtypes here, and I know if you would send out histological slides or paraffin blocks, you would get a full spectrum of diagnosis back from these cases. But from the molecular side of this DNA methylation profile, it was absolutely a massive standard. So none of these 12 centers gave, all of these 12 centers gave back exactly the correct diagnosis for each of these tumors. So we had actually almost nil discrepancy between these centers. And that is very, very different to other laboratory comparison trials that we have seen in the past, where we just always have different schools of pathology and different things. And so here we seem to have a very common language, and that was used mostly here for a certification for a European-wide trial for pediatric brain cancers. But since then we've also certified other laboratories if they wish kind of to join this, this group here of highly standardized diagnostic laboratories. Now, coming to my last point, is a new option that is kind of emerging by DNA methylation profiling, because the procedures that I just told you will take about a week in the laboratory. So if we want to use these arrays from Illumina, it takes us a week until we have the result, which is sometimes a problem and which blocks us from other interesting things. And there is a new sequencing technique which is called nanopore sequencing, sometimes also called third generation sequencing, so the generation after the next generation, which is in principle a very long read sequencing technique, which I will not go into detail here. But the very specific thing which is of interest to us is that it also allows the testing of DNA methylation at the cytosines, so at the cytosine basis, so the information that we require also for DNA methylation profiling. Already in 2017 in Paris, a group here demonstrated that you could use DNA methylation profiling for a one-day diagnosis of brain tumors, back then with a very limited data set. And the first author back then, we managed to bring him to Berlin, and he's now a part in our neuropathological department, and we demonstrated that this indeed works very well. There is limitations of nanopore sequencing. The one thing is that it doesn't work on formalin-fixed paraffin-embedded tissue. You need fresh tissue, so that makes tissue linguistics much harder. But as I said, it's a very strong acceleration of the data generation, and the machinery that you need for the initial testing is extremely low. And it was then also shown that you can go much faster than just doing it at the same day. But there was, for example, this group here in Oslo that demonstrated in 2021 that you could also use this DNA methylation profiling tool for an intraoperative setting. And they managed to generate DNA methylation data and come to the full molecular diagnosis within 60 to 90 minutes for most of these cases. And that was even further explored in this very fascinating manuscript here that combined kind of deep learning with the DNA methylation profiling, and they were able to generate a classifier that allows classification of or generation and classification of these profiles within something like 30 to 40 minutes. This is a group from Utrecht around Professor De Ridder that kind of have brought forward kind of such a tool which is called Sturgeon, which now several centers, also we at the Charité, are testing for an intraoperative setting, meaning that the whole diagnostic procedure, which currently takes over a week, sometimes often 2 weeks, kind of with full classification, may move completely into the operation theater and may then give the surgeon completely different possibilities for decision making already during surgery. And the last thing is that you can also use these techniques and these classifiers that generate, implement nanopore methylation data for the classification of cerebrospinal fluid samples. That is something that if you have a tumor, it will, and if it becomes necrotic, that many malignant tumors are, that it will shed off DNA into the cerebrospinal fluid, and you can then, without operating the tumor, extract cerebrospinal fluid and make a diagnosis from this material. The other setting is that you can also have tumor cells that inhabit the cerebrospinal compartment, and you can then use nanopore sequencing to generate kind of a classification of these samples. This will fail in many cases, so the sensitivity is still a big problem. In 10 to 20% at the moment, I would say that we can make worthwhile information for these patients, but it is a field that is very, very strongly developing, and a lot of people are currently looking into this. And as a last case, here's one example from the Charité, which was a 74-year-old patient with a right ocular nerve palsy for over 8 weeks. Then neuroradiology was done with this lesion here affecting kind of the thalamus and structures below the thalamus. And neurosurgeons were not very keen to operate this because it's a region very hard to get at. CSF cytology was not able to come to a diagnosis here, but was suspicious for tumor cells, and also the FACS analysis of CSF was did not contribute to diagnosis. Here methylation profiling was done, and indeed we could generate a high quality profile of a lymphoma of the brain with very typical chromosomal changes, and that was then also in the future then characterized by a tissue sample from a different site. With that, I would like to thank you for your interest. I hope that it was within the spectrum of what you like here, and I'm very happy for questions at this time.
Any questions in the audience?
Thank you very much. How accurate is this luminometer that you have presented in terms, for example, of selectivity and sensitivity? In other words, are there also false positive results eventually?
Yeah. So this was what I just kind of broke down in this false prediction rate of 1%. So that is, in fact, that is the specificity. So we would say at the moment, the system as it is kind of balanced, we're at a specificity of 99%, which in principle was achieved because cut-off values were introduced into the results that come out of this machine. Therefore, as I said, 12% of the cases you will get the information of I do not recognize this tumor. So this is kind of the sensitivity of the system. which would then be at approximately 88%. So if you ask for these numbers, I would say the way it is tuned, sensitivity 88%, specificity 99%. And it was tuned in the direction of a maximum specificity as it is planned kind of as a secondary assessment after the pathologist. So it's not kind of intended as a standalone technique at the moment, but it's kind of the secondary test, and therefore it was designed to be as specific as possible to not generate confusion kind of at the end of a process, but and therefore we accepted a lower sensitivity for the system.
Questions?
Short question, short one. Thank you very much for this great presentation. We talked about that yesterday, so about the integration of gene methylation in other solid tumors, for example in colon cancer, breast carcinoma, lung cancer. Do you think that in the future we need to integrate some gene methylation signature in our daily practice use or not?
So I think it will remain, for the time being, I think it will depend on tumors where classification is really key. So I think for some tumors, the driving mutations may be more interesting, because you're kind of mostly looking at targeted therapy and these kind of things. And maybe the actual cell that caused the tumor is of less relevance. For other tumors, like for brain tumors or sarcomas, where there's also a system like this, kind of that is in very, very advanced development, where the actual type of tumor is often of high relevance, I think I would strongly advise to integrate it into daily routine. For other tumors, it has to be shown. And I think for many of these, as we also discussed yesterday, there will be rather specific systems that answer highly specific questions. So for example, one that we discussed yesterday, a system has to be created that allows, if you have a squamous cell carcinoma of the region of the oropharyngeal tract, maybe you had that 5 years ago, then you suddenly have a new lesion in the lung. Then it can be very tricky to say if this is now a newly developed squamous cell carcinoma of the lung or if it is actually a metastasis of this tumor several years ago. That is a very, very complicated question in pathology. And for this, for example, such tools have been generated that function extremely well because they can recognize the signature. There's enough differences kind of the cell of origin of a lung squamous cell carcinoma under the oropharyngeal squamous cell carcinoma to make this distinction. So I think it's rather going to be more for specific questions to start with, but there is also at the German Cancer Research Center a big project that will try to have a pan-cancer classifier, so where every type of cancer is kind of included. I think that will take quite a few more years, and it's possible that other techniques will overtake in the meantime. But yeah, so I cannot tell you, I don't think that it's going to be sensible for every type of tumor for the time being.
If I can add just a short question concerning the gene methylation in circulating tumor DNA. Now there is a huge topic concerning the detection of DNA methylation in the blood to distinguish, for example, lung adenocarcinoma versus squamous versus small cell lung cancer. So there is now new technology developed by Warren Health in US. And they claim that it's possible to make the diagnosis of a lung adenocarcinoma versus squamous carcinoma according to the DNA methylation profile. I want to have your opinion on that.
Yeah, we also discussed that briefly yesterday, but this is not fully my field, of course, that we have tested to do also blood testing. So we, for example, did this for prostate carcinoma, and try to evaluate kind of how early could we detect prostate carcinoma from the blood. We were not very successful with that, but it is possible that, the DNA very likely will shed very early, so it's all a question of sensitivity in this context. And how do you kind of get rid of the noise that is just part of our body? So we were not able to do that, but I could imagine that a very focused company would have the possibility actually to really achieve that and to diagnose this much, much earlier than MRIs or histology, for example.
Thank you for the presentation. I have a question about intratumoral heterogeneity. In other words, if you take 6 biopsies and you sort it, will you get a consistent result? Or in other words, is this sort of a branch methylation or a trunk methylation?
Yeah, so this was tested for various entities, and it seems to be that it's not identical for every type of tumor. So for example, for epineumonoma, which is a specific type of brain cancer, we realized that it is very, very stable, also from one operation to the next operation, sometimes with years in between. So that seems to be highly stable. For one of the other very common tumors, like glioblastoma of adults, we do see that this tumor type seems to consist of 2 different subtypes of glioblastoma that can be intermixed within the same sample. So what we don't see is that, for example, within a glioblastoma, if we do multiple samples, we get the profile of something completely different. So it will always just interchange between these 2 subtypes of glioblastoma, but overall we do see that, of course, the filtering and the generation of the machine learning tool kind of very, very, very strongly favor branching stem features kind of DNA methylation, because we just filter out a lot of the non-usable information during the process of the machine learning calculation. Sorry, that was maybe a bit complex in the end. Yeah, anyway.
Another question? Okay, I have a last question. Coming back to the morphology.
Oh yes.
So the visual classification of brain tumor is a little bit different compared to the other visuals, because we have this general grade of malignancy, grade 1 to grade 4, indolent to high diagnostic tumor. Using your classifier, what do you think about this a little bit artificial grading? And are we to continue to grade with 1, 2, 3, 4?
So the methylation doesn't see grades, so that's quite clear. So if you look from this biological perspective, the system is nearly blind for kind of different WHO grades within an entity. So it will just give you the information of this is your entity, and so I think that a lot of the grading of the past was also because we had problems to separate the boundaries between our tumor classes in the best possible way, so that we had intermixing classes and that this influenced kind of outcome statistics quite a lot. So to us, the data now looks much cleaner, and I think grading will be of less importance in the future. I cannot tell you that you will not have to do it anymore, but I think it's going to be of less importance.
It's very easy for clinicians to have this grade.
They love it. Yeah, that is of course true. I would say pathologists, neuropathologists usually fear this grade because we know the subjectivity that also goes into it. Clinicians often like it because of course it's very clear this information of how to treat a patient.
Okay. Thank you very much.